And now we continue. The last but not least, Khuram Chaudhary is joining us, also from the US. I don't think I need to introduce you a lot, Khuram. Everybody knows you very well. What I can say is that after many years at GSK, you are now Scientific Senior Director at BMS. So congratulations for your new adventure. Thank you very much for joining us. And today, Khuram will have a very nice overview about the results that have been obtained on this various platforms at HESI Stem Cell Working Group, talking about chronic drug-induced cardiotoxicity assessment of using human iPSC-derived cardiomyocytes, and what is really interesting, also human heart slices. So I'm very excited for your talk. Thank you so much for joining. The floor is yours.
Can you hear me okay?
Yes.
And is my screen visible to everybody?
To me, yes. So I guess to everybody else as well.
Okay, very good. Thank you again for the invitation. Let me just cue everything up. Okay, so I want to thank Nana and my friend Sonia for this invitation to speak at the symposium. There's some very interesting talks we've heard already today. I'll be providing an update on a global collaboration that, including many of you who are on the call today, under the HESI umbrella, which is the Health Environmental Science Institute. Our goal is to assess chronic treatment-induced cardiotoxicity in preclinical development. And that's something that I think many people have worked on, but in this collaboration, we're really coming up with a better way to do the preclinical development. So let's see, okay. All right. So to begin, I'll share a little background on drug development and the causes of failure that we see in non-clinical and clinical development, the mechanisms associated with long-term toxicity that go beyond ion channels alone, which is the standard ICH-S7 supportive study. And we'll follow that up with a very high-level update on stem cell working group collaboration. So as part of the background, we all know that drug discovery and development is an exercise in failure. We have very few lead molecules early in discovery that actually make it to the clinic. But what we know is that the cost of failure becomes exceedingly high if that failure occurs later in development. And so it's sort of a paradox there. The major causes of attrition haven't changed for decades, and they include more than half of that attrition due to efficacy and human and animal safety and toxicity. And over the past decade, regulators and industry have emphasized the investigation of ion channel dependent QT prolongation. As you know, many of you have talked about SIPA today, as well as what you've heard recently over the last year in terms of the ICH-S7 E14 Q&As and the best practices that were adopted. However, adverse drug reactions often occur or are reported for cardiovascular events, including heart failure, myocardial disorders, changes in blood pressure, structural changes, including fibrosis, as well as vascular injury. And so it's not just a matter of understanding the acute cardiotoxicity, rather what might happen over chronic treatment with respect to function and structure. Designing the preclinical strategies to assess that risk, though, can be very challenging, as most models require the use of animals dosed chronically over time, and then sort of a retrospective analysis of what mechanisms were modulated. So here I've cited a recent review article that identifies various mechanisms for drug-induced cardiotoxicity. And what you can see is that that goes beyond just ion channel pharmacology. These include signaling pathways, growth factors, energetics and mitochondrial function, apoptosis, ion channel trafficking, and changes in mechanical function, including fibrosis, as well as we mentioned earlier, electrophysiological changes. As you can see, there's multiple drugs that have been withdrawn from the market due to cardiovascular toxicity, and that goes beyond just the ion channel and QT prolongation. And these drugs actually span therapeutic targets and organ systems. So it's not just the cardiovascular specific therapeutic area or indication that we're concerned about, but it's drugs that are, for instance, anti-neoplastics, anti-cancer agents, GI as a target, anti-inflammatories. So it goes well beyond just understanding what happens in the setting of cardiovascular disease as a target indication. So the advantage of working in a partnership like HESI is that they're able to bring together industry scientists, regulators, academicians, and CRO partners to address a specific concern. So the goal of the stem cell working group was to identify relevant endpoints that could translate to clinical cardiotoxicity. We also had the goal of selecting an appropriate reference set of compounds with specificity to benchmark these endpoints. And you can see those here. There's about a dozen here. And you can also see the endpoints that we were interested in capturing. We wanted to test the hypothesis that might bring up a signature or a fingerprint of chronic treatment-induced cardiotoxicity, including both structural and functional tox, using non-clinical in vitro assays. And so what we've done is engaged with greater than 20 different partners across industry, academia, regulatory, and CRO contributors. And the study design for this was actually quite prescriptive. So the goal was use similar reagents, capture the data at similar time points, whether that be 1 hour post-treatment to understand the acute cardiotoxicity versus 24, 48, 72, and so on. But also then to look at different endpoints across these platforms. And so what I'm going to do now is share with you some of those technologies that we used, especially some of the more innovative ones. So here, we have a subset of technologies and reagents employed in this collaboration. This helps illustrate the breadth of the effort. First and foremost, the goal was obviously to use in vitro and ex vivo systems, including stem cell-derived cardiomyocyte monolayers. You can see that here, engineered heart tissues, as well as using primary human myocardial slices. And so when you look at some of the endpoints captured here, you're probably relatively familiar with them. And those include microelectrode arrays, contractility measures for monolayers, changes in action potential using fluorescent indicators, actually measuring physical force and contraction from the tissue preparations. And so again, the durations and the sampling intervals were very prescriptive, but each individual contributor was able to use their systems however they saw fit, obviously to manufacturer specifications or to some level of maturity that we could say was steady state, and that was representative of cardiac physiology. So I'm going to further illustrate some of those technologies by showing you examples of things that are interesting and kind of span the chronology of drug development from that early hazard identification through to looking at signaling cascades and mechanisms. And so here, this contributor suggests a representative trace from a bright field imaging platform. We have an instrument that captures that bright field image from a dish of a monolayer of cells. It uses landmarks associated with bright field imaging from those cells. And as that landmark or the speckles change, that deformation is picked up as a waveform. So that waveform then allows us the measurement of relevant endpoints such as beat rate, the contraction velocity and relaxation velocity, amplitude and relaxation parameters. And as you can see, these measures change with chronic exposure. And in this case, we're looking at doxorubicin, which is one of our positive controls, as well as in a concentration dependent manner. And so we hope that the results here mimic what we see in terms of progressive cardiotoxicity and a decrease in function that's observed in patients who are taking doxorubicin or other anthracyclines, which we know cause a progressive reduction in cardiac contractility. So this slide is a little bit more animated, so I'll run through all these different steps. In this assay, it's a plate-based assay with higher throughput. We use stem cell cardiomyocytes that are cultured with maturation stimuli. So in this case, the extracellular matrix has various factors that enhance maturation. You can see on the top right, you get a very striated sarcomere pattern. You also get binucleation of the myocytes, which again suggests some level of maturation. And so using this high throughput system, you can actually, can't run the, oh, for some reason I can't run the video. But what you'd see here in the 96 well plate is that the entire plate flashes, and those flashes are a measure of the calcium transient as picked up due to a transduced protein called GCAMP. This is a calcium indicator protein. And so in real time, we're able to measure that calcium signal, which is a surrogate of contractility. And you can see that as you capture that over time with your concentration dependence, you see some progressive decrease in the calcium homeostasis. So again, this is a measure of that progressive drug-induced chronic treatment induced cardiotoxicity. So we also employed many of the technologies you're familiar with in assessing acute cardiotoxicity. And that includes the microelectrode array, an impedance measure for contractility, as well as novel assays such as the Flexite, which culture cells on a flexible membrane and actually allow us to measure contraction. So again, I'm very cognizant that we highlighted the sampling intervals because again, we want to make sure that we're capturing our data in a sort of a harmonized way, even though the assays are very protocol specific and unique for their endpoints. And so here's some of the representative traces. On the left, you can see there's field potential shortening due to the exposure of this compound. And that sort of falls in line with what you would expect in terms of contractility. Oftentimes, calcium channel blockers will shorten the field potential duration or action potential duration, and you'll see the decrease in amplitude in your impedance measure. And you can also see that over time in your Flexite. And so these assays are confirmatory of each other. And so now not only are you looking at one endpoint, but by measuring several endpoints in parallel, you can actually come up with a signature or sort of, like that chicken or the egg phenomenon, like which mechanism is actually leading to the depressed function. So as we move on, we're looking at more complex outputs, right? So in this case, the contributor was measuring field potential duration as well as that functional change, but then using the Luminex to actually measure biomarker release. So we have an assessment of biomarkers such as troponin, creatine kinase, BNP, over the different treatments, and that allows us to assess cellular damage. So if you look here, you can see some of those mechanisms of changes in contractility that you'd see over time. So we have creatine kinase release, FABP3, BNP, NT-proBNP, and that's measured over all of the different treatments over time. And so in this case, we saw that biomarkers such as BNP were released in troponin, and that's exactly what you would see in a clinical setting, right? So the first indication that you have some type of cardiac dysfunction would be a measurement of troponin as a biomarker in your bloodstream. And then you'd go on and do electrocardiogram as well, and then sort of get to the root of whether you had an infarct or whether there was some progressive disease happening in the heart. And so this is the type of thing that we don't often see until late stage in vivo studies. So finally, we hope to investigate the mechanisms by which chronic toxicity occurs, including metabolics, energetics, mitochondrial function, and contributors here were able to measure metabolite production, such as lactate and arachidonic acid. We also looked at microRNAs, which are sort of a newer biomarker for cardiac disease, knowing that these, when measured by PCR, are indicative of cardiac damage. And this is just a sample of that data. Again, I'm not going to go into too much depth on this because the results are just now coming in. We have a statistician who's working to collate all of that. And then we're working very closely to design that fingerprint, as you might call it, for that preclinical development signature. And so if you look at this data, what you have is concentration dependence in the various biomarkers and metabolites, also the microRNAs. And again, we're looking at that over time. So the next thing I want to show you is how you take that functional endpoint as well as the biomarker and come up with some sort of signaling pathway or mechanism that explains why you have that progressive change in function. So in this example, the contributors are using the seahorse assay to measure mitochondrial respiratory capacity as well as stress, and that includes oxygen consumption in parallel to the functional endpoints. So analyzing the mitochondrial oxygen consumption in parallel to those functional changes gives you that mechanistic approach and a signature to the compound-induced toxicities. So you can imagine that when you take this and you develop some sort of heat map or some sort of principal component analysis, you can actually come up with not only the progressive change in function over time in vitro, but a potential mechanism by which that occurs. And so additionally, the measurement of mitochondrial membrane potential provides further insight into the compound-dependent effects that may create things like reactive oxygen species, things that may inhibit the electron transport chain, leading to progressive changes in function. And so we hope to create a signature which you can differentiate that chicken or the egg with respect to the cause of the cardiotoxicity. And that may otherwise only be investigated retrospectively after you've seen something in a late stage development study in vivo. So in summary, while significant emphasis has been placed on the non-clinical assessment of acute cardiotoxicity, we've all heard about the ICH S7 Q&As and the E14 best practices. Less is known about the chronic treatment-induced effects, including structural cardiotoxicity, prior to late-stage in vivo studies. So the goal of this project is to establish that predictivity, sensitivity, and specificity of various platforms that use novel reagents like stem cell cardiomyocytes, as well as primary human cardiac tissue as a benchmark, ultimately to understand what happens in terms of chronic actions of drugs. And so before I conclude, I want to make sure that I thank all of the contributors. You can see there's a wealth of knowledge here. There's a multitude of contributors across industry, academia, regulatory, and CROs. On the right alone, you see we have over 20, upwards of 24 different contributors working on this. It's been a global project, again, supported by HESI. And I think we're at the point now where the results have come in. We're at the point of doing statistical analysis. There are novel ways to visualize that data, and you should be hearing something at various conferences coming up. We started with SPS last year. We tried to do a presentation at SOT. There's some work coming around for Safety Farm Society later this year. And you should see some output from this as part of the stem cell working group. So I want to thank you for your time. I appreciate the invitation, and I'd like to open it up to any questions.
Thank you very much, Khuram. This was wonderful. I enjoyed a lot. Do we have any questions? Anyone writing anything in the chat? If not, I can ask two. We have one from Sonia, actually. So she's asking how far the analysis of the data go. Can we expect a publication soon, maybe?
Yeah, that's a good question. We have a sub-team that's dedicated strictly to the data analysis. You can imagine that while each platform is different, we were very prescriptive on how the data should be submitted. So we had a group come up with a table format where we divided up various endpoints, the time points, each assay parameter. And so just on sort of field potential alone, there's 3 or 4 different endpoints. And you can imagine across the various platforms, there's a multitude. And they're all different. So we're trying to harmonize that. But we've got a really good team of statisticians and scientists working on this. I've seen some of that analysis already in heat map format. They're writing some scripts on how we can automate that analysis. There's a sub-team of individuals who volunteered to do that analysis as well. So I wouldn't be surprised if you see something within the next year in terms of a publication.
Well, that's good. Very fast track, I would say. And I have one for you, if you don't mind.
Yeah.
This is a very comprehensive study, enormous amount of work, a huge team. What kind of insights did you personally get from all of this data that you have performed? What would you think would be the most interesting that you have seen so far?
Yeah, I think we took a very agnostic approach or an unbiased approach to coming up with the assays, reagents, platforms. So no bias going in to say, well, like stem cell cardiomyocytes are immature, or we only need dog or monkey tissue, or we only need human tissue. And so when you take that approach, what you see is there will be endpoints that are not sensitive. There will be reagents that are not sensitive, and you'll find those that are. And so the goal here is not to be that prescriptive. It's one, to assess the field and say, in vitro technologies and novel approach methods have some utility in assessing chronic cardiotoxicity. This has a potential impact on 3 Rs, so reduction of animal use, being more predictive of the clinical setting. So as long as you're open to all that, you'll find some assays will be more sensitive, some will be less, and it'll really help us to design a more appropriate, clinically relevant strategy early in drug development.
Thank you. And now from Ron one, it starts a little bit funny. Without having a crystal ball, do you think the regulatory authorities will incorporate the newer assays, and when?
So I think it's a really important distinction. Much of the work that we're doing in HESI is meant to be exploratory, investigative, and to help people better understand how to make safer molecules for patients, right? Unmet need. There's obviously a regulatory component because they are our partners. We want to ensure that when we submit this data, it's done in a manner that's relevant and they can interpret it. I think there's quite a bit of buy-in from regulators. In fact, if you look at the concluding slide, there's several individuals from the FDA who we've partnered with who are generating some of that data as well. I think everybody's familiar with the FDA Modernization Act. So again, there's a culture right now of shifting to novel approach methodologies, organs on chips, microphysiological systems. And I think that's the future. So I think there's quite a bit of regulatory buy-in.
Okay, from Katerina, one more. Hi, great presentation. She has two questions. What is the first time point you consider as chronic one? And have you already published some protocols for the chronic cardiotoxicity screening if someone would like to follow it in their studies?
So let's start with time points. What you'll probably intuitively understand is that this is not an in vivo setting. And so we don't have the same drug disposition properties. We don't have the same metabolism, the clearance. And so, a PK profile in an animal would look like, the sine wave. Here, we're just adding compound every 2 days. We add fresh compound and we change the media. So it's not truly a mimic of an in vivo study. It's really a matter of having that chronic exposure and eliciting signaling pathways that could be detrimental over time. So it's hard to say what is a 1-for-1. Is a 7-day rat tox study the same as a 24-hour, in vitro treatment? I don't know. Again, this is hazard identification. So that's a tough question, but what we've chosen to do is work within the boundaries of the reagents as well. So these stem cells, for instance, have to be fed every 2 days, or we like to think that they're fed every 2 days. We're also using serum-free and serum-containing conditions. That has an effect on the physiology. But every 2 days, we replenish the compounds, and we've gone out to 96 or the next 24-hour increment, so up to 4 or 5 days in terms of looking at the effects. Again, it's really a matter of seeing that progressive change, and that's the hazard ID. So that's a generic way of answering your question. And so the second part was how much of it's published. Well, keep in mind, we're trying our best to stay within the protocols or the methodologies that have been given to us by the reagent manufacturers and the assay platforms. And it's a matter of doing things in that controlled prescriptive manner, but looking over time. We're trying not to change any of that culture condition just to see what the sensitivity would be. Let's just see what the output's going to look like. I don't think we've done this in a concerted way like this effort ever. So that's yet to be determined.
Thank you, Khuram. And one more from Stephen. Is it reasonable to assume human iPSC-cardiomyocyte would model human cardiotoxicity in response to drug versus isolated mouse cardiomyocytes?
I think that we're going to have a conversation over dinner or a coffee. It's a very long answer. I think there is utility in using primary mouse cardiomyocytes, but I think we all agree you can go back to the early 1900s and find that the physiology of the mouse and the human are very different. The calcium homeostasis is very different. The acute function is very different. EC coupling is very different. EC coupling gain is very different. So I can go on and on and on, but it's a living organism. It responds to drug treatment, and it's often a preclinical species, non-rodent or preclinical rodent species used in drug discovery and development. So there is utility. I think what we're learning is that if you go just to that pie chart I showed you, one of our biggest problems with drug development is efficacy. Is it because we're only testing on mice? Possibly. And so the human relevance, I think, is very important. And what we're trying to do is establish human relevant assays that have human proteins, that reduce the use of animals, and that give us better clinical translation. So there's a place for all of this.
Very nice way to conclude the meeting, I think. And thank you, Khuram, for your talk and for the discussion. I will now, for the closure of the meeting, just share a couple of slides from my side before we say goodbye for this year. So I will try to share my screen now. Let's see if that will be possible. So I hope you see my screen in full view. Yes, so just to maybe raise a little bit of attention of some news or changes in our image that you might have noticed lately. So we have a new website that we've launched 2 months ago. And I would like to invite you to visit it and maybe look at these 2 areas here. So the application areas and therapeutic areas. We have tried to finally curate information, not only from us, but also from other scientists for various, for example, disease models, for various application technologies, and so on. And one of them is cardiac safety. So we have a whole page dedicated for cardiac safety, which I would invite you to visit. And before we all leave this meeting, I would also give a little bit of shout out to other meetings that are coming. One of them is not organized by us, but is approaching rather fast. The deadline's actually, I think, on 10th of June already for the abstract. If you're all coming to Safety Pharmacology meeting in Brussels this fall, please join us. We will have posters, we will have our booth there as well. But also in October, we are hosting our annual user meeting here in Munich from 24th to 25th of October. You're all kindly invited to join us. And at the end, I would like to thank you, every one of you who joined, especially our speakers. These talks were wonderful. They covered a lot of topics. And I think our speakers joined us as well as you attendees from, I think, every possible time zone. So thank you very much for staying late, waking up early. These were wonderful talks. Have a wonderful day or evening, wherever you are. And hopefully we see each other in 2 years for Cardiac Symposium 2025. Thank you very much. And we will follow up with recording of this video, so everyone who registered will get a copy. Thank you very much, and bye-bye.